let's start. Till now we have studied about the shape, size, reproduction, basically all the physiology in the bacteria, everything is done. Now it's time to discuss how we will take the bacteria from the sample, okay, and grow them. We already seen in yesterday class about the growth curve, that bacteria shows you can say different stages in their growth and development. We also seen that in the bacteria there is a one ways by which bacteria can grow their population. That's a binary fission. We discussed that growth and what is called as the multiplication in bacteria. Now in today's class we will discuss okay fine, naturally the bacteria is present in the different organisms body or either in the dead decaying substances, fine. So if it is present in the human body, then we have to take the samples from the human body, fine, and in that sample there is a very minute bacteria is present, a very small amount of bacteria will be present. But in order to see those microorganisms, in order to establish any significant, you can say, diagnosis, or if we want to label the bacteria, we need a proper, you can say, description about the bacteria. So for proper description, we have to grow at a larger amount. So for that growing purpose, we have to choose the culture media. We have to choose the culture media. Means basically we will take the sample, okay, we'll take the sample from either the human blood, okay, or else basically the serum, okay, the sputum, urine, fecal matter, okay, or also called as the fecus, fine. Then what else? We can take the samples of CSF, cerebrospinal fluid. Have you heard this term? Yes. So from all these things, we will take in the sample, fine. We take in the sample, and these samples will put into the a test tube, okay, test tube. Then from this test tube, we will put the bacterial sample into the nutrient media via the inoculations, fine. Now this nutrient media is mainly the two types. First classification is based on the physical state. The first is the solid media, liquid media, and semi-solid media. We have the three classification under the based on physical state. Then we have based on the function. Based on the functions, the media are how many types? 6 types. How many types? 6 types. First one is enrichment media. Second one is the enrichment media. Third one is the selective media. Fourth one is the indicator media. Then we have the differential media and transport media. Fine, because bacteria are it is the top most abundant organism on this planet, fine. And as I said you that as we choose the food according to our preference, we want to eat this, we want to not eat this. We are the selective in nature. Bacteria do follow this selective nature, okay. Bacteria are also basically they prefer to eat something and do not eat something. So for that we have this many culture media. So what is culture media? It is basically a mixture of different chemicals. What is culture media? It is basically a mixture of different chemicals which provide basically support to the bacteria. Support, it means support in terms of their growth and development. Fine. Now the another thing which you have to keep in mind about the culture media is that it is the very crucial, or you can say very important for the laboratory. Without this culture media, you cannot see the microbes. Because you will take the limited amount of blood, maybe the 5 ml of blood you taken from any person. Fine. In that 5 ml also you have the very little, you can say the very small slide. You will put only some amount of blood. Fine. But in order to see, we need more bacteria. We need more bacteria. So how we will produce the more bacteria? One way is to provide the nutrition. And nutrition is coming from the culture media. Clear? Yes? Fine. Now coming to the first classification based on the physical state. We already know that physical state consists of the three things, either the solid or liquid or semi-solid. Why not gas? Because gas will not, for gas we have to enclose the vessels or enclose the media plate, but that is not needed. Fine. So first example is the solid media. Solid media. Solid media, the same things happen. Fine. We taken the sample, put it into the test tube, then inoculated into the where? Yeah, into the media plate. Now, this all medium is what? Physically, they are all the liquids. They all are liquid. Your blood is liquid, serum is liquid, sputum is semi-liquid, okay, and the urine is liquid, fecal matter is also a semi-liquid, and CSF is of course a liquid in state. But the thing is, it is a solid media. So we have to basically make it solidify. So in order to make it solidify, we have to add certain chemicals. Have you heard about the agar? Yes. We will add many things here. Okay, we will add many things here. Starting from first, we have to provide here the nutrition. This is a simple media. Okay, nothing is added. Just only a petri dish, like a small boil. A petri dish. This is called as the petri dish. But we have to make it a specific nutrient media. We have to make it a specific culture media actually. So in order to make it a specific culture media, we have to add certain things in it, which basically support and grow the bacteria. What we need is the carbohydrate, protein, fat, vitamins. This is what we need for our growth and development. What bacteria need is a source of protein first. First thing they need is a source of protein. Second thing they need is electrolyte. Third they need is water, okay, and that what is called as a solidifying agent, agar. So first we will add in this is the peptone. Now what is the peptone? Peptone is basically partially digested protein. It is a partially digested proteins, fine, which is basically a source of protein for the bacteria. So first we will add the peptone, water. It is also liquid in nature. Like we need protein, no? So for protein, what we are eating? We are eating the pulse, dal hum log khate hain, and we will add like paneer and all the stuff. That is for our source of protein, fine. So for bacteria, we will add the peptone. Peptone is nothing. It is basically partially digested proteins. Protein, which is basically liquid in nature. In market you will see it is sold in one tube. Okay, एक ट्यूब वाला डब्बा में आता है, दुकान में वो मिलता है, fine, and that is basically a source of protein. Got it? Now the second things which we are going to add is the NaCl. What is NaCl? Yeah, sodium chloride. It is basically a electrolyte. Fine. Then another thing which we are adding is, yes, another thing which we are going to add here is the water. Basically not a normal water, it is a distilled water. Fine. So we are adding here a distilled water. Now the four things we are going to add here is the meat extract. Basically the beef meat. Which meat? Beef meat. Now what is the use of this meat here?
Source of protein.
Yes? Source of protein. For that we provided already the peptone. Carbohydrate is not that much needed. Basically the meat extract is basically providing the whole, you can say whole bunch of nutrient. It do have the protein, but specifically we are not providing for the protein. Specifically talking about fulfilling the protein requirement to the bacteria, we have the peptone. We have the peptone water. This meat extract is basically source of vitamins, is a source for vitamins. Fine. Bacterial vitamin need is fulfilled by the meat extract, okay, and other growth factors, and vitamin A. Okay, vitamin A, this both the things is, you can say, provided to the bacteria by the meat extract. And which meat? Beef meat generally. No problem. Now, talking about the last constituent, what is used in order to make the culture media is that water. See, these all are basically water we already added, no? Yes, distilled water we added, meat extract we added, NaCl added, and peptone. That's all. That's all. This all thing is, and yeah, the last part. These all things is liquid in nature, isn't it? Your peptone water is in the liquid, NaCl is in liquid, distilled water is of course a liquid. Meat extract we are giving is in a powder form, okay? It is in the powder form, but generally it is also comes under the liquid forms too, okay? But these all are the liquid in nature, but then we have to solidify it. ये सारा liquid के स्टेट पे है, बट हम लोग को इसको क्या करना है, तो इसको सॉलिड करना है, ओके? तो सॉलिड करने के लिए वी आर प्रोवाइडिंग द agar. Agar is widely used in the field of microbiology and more specifically in the lab. Why? Because it solidify the things. Solidify the things means basically ये जो liquid, सारा चीज liquid है, इसको क्या कर देगा, जमा देगा, इसको solid बना देगा. एंड ये agar हम लोग बेसिकली एक्सट्रैक्ट करते हैं एक समुद्री घास से, और seaweeds, okay, called as the Geladium species is a grass species, okay? ये घास का एक जाति है जिससे हम लोग what बनाते हैं, it's a agar. Okay, agar, Geladium. Clear? No problem. Now, one thing you have to keep in your mind is that these all the constituents, peptone, sodium chloride, distilled water, meat extract, as well as the agar, this all is used in throughout all these media. It is also the same chemical constituents of the solid media, same for the liquid and same for the semi-solid. The thing is, we are only varying the proportion, the percentage. We are only varying the percentage. Got it? Sometimes we are using the 2% agar in the solid media or 5%, then we will use in the liquid media a less agar. Okay? Clear? No problem? Understood? And example is the blood agar and the nutrient agar. We will see when we will discuss the based on the function, then I will teach you the blood agar in bit detail. Semi-solid, the same things. Example is the thioglycerate broth. Have you discussed this thioglycerate broth previously? You remember? Yes, we discussed, no? In the differential media we discussed aerobic and anaerobic.
Yes, sir.
Yes? That aerobic bacteria will grow at the top of that, and then we have studied, no?
Yes, sir.
Yes, when we are dividing basically the bacteria on the basis of oxygen requirement, on that time we discussed about the thioglycerate broth. It is nothing, एक ट्यूब लेते हैं, ट्यूब के अंदर में हम लोग क्या करते हैं, तो कुछ केमिकल्स मिलाते हैं, और केमिकल मिलाने के कारण क्या होता है, तो ट्यूब में जितना भी ऑक्सीजन रहता है वो सारा गायब हो जाता है, उसके अंदर में हम उसके बाद उस बैक्टीरिया को रखते हैं. बैक्टीरिया के रखने के बाद जो बैक्टीरिया एरोबिक बैक्टीरिया होगा वो ऊपर में ग्रो करेगा, जो एनारोबिक बैक्टीरिया सब होगा वो नीचे ग्रो करता हुआ जाएगा. दैट इज कॉल्ड एज द thioglycerate broth. एंड वन थिंग यू जस्ट रिमेंबर, कीप इन योर माइंड, इज दैट इन द माइक्रोबायोलॉजी broth means liquid. So either we can use the thioglycerate liquid media or thioglycerate broth media. No problem. Coming to the semi-solid media, and these all have the different use. Solid media is used for understanding the bacterial shape and size. Okay? Solid media is used for the shape and size. Liquid media is used for understanding the bacterial, you can say the characteristics, like more deeper characteristic, their growth pattern, okay, their growth pattern. Semi-solid media is used for understanding the motility of the bacteria. Okay, it is used for understanding the motility for the bacteria. So for exam point of view, you have to keep in your mind the three things of each of the medias. If you're talking about the solid media, you have to understand what is actually the solid media. Okay, and the thing is, the solid media is made up of these all things, the peptone and all these things. This is the constant. So one time you just remember and you will answer all the questions. So first thing is, what is the solid media, you have to understand. Okay, second thing you have to understand is the examples. Okay, examples you have to remember. They will ask maybe in the MCQ that example of solid media, they will put blood agar, thioglycorate, and these two examples you have to choose the right answer. Okay, then you have to also keep in your mind about the each of the media is their what is called as the use. Okay, their use. They will ask that blood agar is used for, okay, or which bacteria will use the blood agar. I will discuss in a few minutes. Okay, so three things you have to keep in your mind for exam point of view. These questions from the culture media, they may be asked from 2 marks or the 3 marks. Okay, no problem. Now coming to the another classification of culture media is the based on function.